Morning, everyone. Thanks for joining. We'll get started in just a few minutes. I think everybody will give it just 1 or 2 more minutes before we get started. All right, it's 2 past the hour, so we're going to go ahead and get started. Welcome, everyone. Thank you so much for joining our Variant Scientist Network event for today. Just a couple of announcements before we get started. The newest issue of the Variant Scientist Network newsletter is live now. If you've not yet joined our LinkedIn group, the group number is listed on the slide that you should be seeing. Go ahead and sign up to join the Variant Scientist Network LinkedIn group for that newsletter and other updates that occur. And then we also send out periodic updates with events such as today and other things to be aware of. And if you would like to sign up for that or for our Slack channel where we have a little bit more informal discussions about variant scientists and issues that affect variant scientists, you can sign up for those updates at the link posted there. And I'll post both of those in the chat as well. The format for today's event is going to be somewhat unique. We're going to start off with a presentation of a Journal Club article by Jasmine Baker, who will be presenting a publication about variant interpretation and training for variant scientists. The first 15, 20 minutes or so will be a discussion of that publication. And then what will follow is a panel discussion of the publication with people who are involved in the training of variant scientists and the hiring of variant scientists. And these will be our panel presenters for today, we have Barbara Fortini from the Keck Graduate Institute. We have Brian Gall from Neogenomics. We have Ashley Birch from GeneDx and Kevin Bowling from Washington University in St. Louis. And with that, I am going to turn over the presentation to Jasmine Baker. Go ahead, Jasmine.
Awesome. Can you enable sharing for me, please?
I hopefully can.
And if you can share it, that'll work too.
Okay, let me see here. Okay, I am not sure how to allow you to present. If anybody knows on Teams how to allow someone to present, I would love to hear about it. Okay. Well, worst case scenario, Jasmine, I will just share the publication itself. Oh, do you have a way to request that?
No,
Jasmine, you don't see it anywhere? Okay.
no, it says only meeting organizers and presenters. So maybe in like the actual meeting settings in Teams, you have to change it or something.
Yeah.
Um,
Okay. Let's see.
okay, usually find the person in the list and right click. Okay.
Okey-doke. All right, I'm just going to share the publication for now and I'll keep trying. I'm so sorry.
Okay, it's okay. Do you have
I have.
the email with the slides and you can just like if you just click through, I can
Okay,
talk and it'll be fine. Yep.
great. Okay, I'll do that.
Alexa, did you click on the three dots on Jasmine's photo to see if you could grant permissions for?
I did, and that
Okay.
only allows me to enable her camera and mic and spotlight her.
Ah,
Um,
okay.
I did try that. Okay, and Jasmine, you sent me the slides?
Yes, so I sent them yesterday afternoonish. I believe everyone was on the email who was on the previous meeting, so you should
Okay.
be able to access it.
I think I can also share, and I have them up if that makes sense.
Would you please do that, Barbara?
Alexa,
Thank
Oh,
you
sure.
so
awesome.
much. I appreciate it.
Okay,
Thank you.
let me get to the first slide, and then hopefully I will be able to share here.
Awesome, awesome. So good morning, afternoon, evening, everyone. So today I'm presenting the Journal Club on variant interpretation training for the genomics era. The paper we're discussing focuses on variant interpretation as more of a professional skill and specifically on how to define consensus-based learning outcomes for people who are doing variant interpretation work. I really enjoy reading this manuscript, and I hope you guys do too. I think it's unique in the sense that it's more focused on the educational standpoint of variant interpretation. And it really tries to address a gap um, in our field. So I know we talk a lot about how to classify variants and like the guidelines like ACMG, AMP, and how to support that process, but we really don't talk a lot about how we train people who are actually doing this work. So they really asked the important question of what should a competent variant interpreter know, be able to do, and how can that be taught at a scalable and standardized way? Um, so as I go through the presentation, I'm going to cover why this is important, how the authors built their educational framework, what the learning outcomes look like, and how they evaluated everything. Next slide. Okay, so lots of summary words here. Hopefully you guys like words. I know some people like to read and some people love images, so there's a mix here. So to start, the paper frames variant interpretation as a professional skill rather than just a technical skill, which is really important. So variant interpretation sits at the kind of like the interface space between sequencing data, clinical decision making. So the downstream consequences are really significant, especially for patient care. If interpretation is inconsistent, incomplete, or if you can't reproduce it, you may have an effect on diagnosis, management, or ultimately patient care. So the authors really tried to emphasize that while genomic medicine has expanded rapidly, the training has not developed in a equally standardized way. So training is being developed, but it's just not standardized. Um, and I think even our group that we have here today, I'm pretty sure if we did like a little popcorn roundtable, people come from a variety of backgrounds. So we have laboratory scientists, clinical geneticists, um, bioinformaticians like me. I'm traditionally trained in computation research, and some people just learn on the job alone. So that means you have a really major variability in how evidence may be gathered, weighed, or applied just because of, uh, the user's just typical way of thinking about how to assess the situation. Another key point, which is on the more on the right side of the screen, is that guidelines alone are not enough. Guidelines give us a framework, but it doesn't necessarily define the curator's competencies, career stages, or their benchmarks for proficiency. So, in other words, it helps us tell like what the classification categories are, but not necessarily how to train a person to become consistently good at interpretation, especially if you have different scenarios. Because, sometimes you get rare diseases and things that are just trickier than others, or they're not in every population. So this study really tried to solve like, hey, how can we fix this educational gap and how can we develop materials or trainings to kind of eventually make more of a structured um, educational resource for variant curation as a workforce, which is really nice because I do believe variant curation is going in that direction as more of this is a career, this is a workforce there, a variety of people. So, um, next slide, please. So next, I'm going to review the methods. So I just kind of broke it down in 4 little points. So the methods are really interesting because it's not a traditional wet lab or computational paper. It's really education. So they focus a lot on instructional design, which may or may not be familiar to everyone. So they use an outcomes-based design. So that just means that they started by saying, okay, the learner should know this. They should be able to do this rather than starting with a course and filling in the gaps in the content. So they took their outcomes and they aligned it to a taxonomy called Bloom's so that the progression could move from foundational knowledge at the base and go to a higher order of application or reasoning about the topic. They made sure that there were experts who co-developed the framework, which is really important. So it makes sure that they kept their learning outcomes tied to real world labs and curation workflows. Um, and they really tried their best to make the outcomes relevant to day-to-day work of people who identify, curate, and actually classify variants, which was really nice. It was also really important that the training that they delivered was multimodal. Um, I I thought this was really nice, especially, in this kind of post-COVID era. It's nice to have things that are self-directed online as well as like blended, so workshops and things that you can also reference, which is also important for like scalability. Different institutions, learners, some people allow different types of formats. So that was really nice to see. They also used a framework called the Kirkpatrick Evaluation Framework to assess the actual impact. I really thought this was important to mention, especially so I'm not sure if some of you guys have done like educational manuscripts and things before, but whenever you submit to these types of journals, they want you to try to quantify it as best as possible. So I thought it was nice that they did use this type of evaluation framework. So they not only looked at whether the learners liked the program, but they also looked at whether they learned something, whether behaviors changed, and whether they had like a broader like impact in regards to like their foundational knowledge in, the individual subcategories of learning. So personally, I thought their approach was very structured and rigorous, especially for a education type manuscript and structure. Next slide. So this particular slide gets into like the central design of the study, which is outcomes first, which we already talked about. And it's really one of the strongest aspects of the manuscript, in my opinion. So instead of asking, hey, what lectures do we need to give? They said, what should a variant interpreter be able to do at different stages of development? So that kind of shifts made the curriculum, in my opinion, more intentional and more in line with day-to-day practice. I'm gonna try to blow up the image on my side so I can kind of step through it. So on the right side, I have a screenshot of figure one just in case you don't have the manuscript in front of you. So they did break things down into what we talked about previously, where they had the self-directed online courses and they also have the in-person workshops. And they also broke it down into introductory and advanced. And there's a scale immediately under it where it gives you the amount of people. So N equals 392, where they did the baseline survey, the completion survey, and they kind of give you these checkpoints of things that happen along the way to make sure they were actually evaluating the users of the content. So registration data, quizzes, knowledge checks, workshop polls. And these learning outcomes that they had, that they evaluated, they made sure that they were refined by the panels, which was really nice, who had that scientific, clinical, and educational expertise. Um, let's see. The outcomes were also organized across three main stages of variant interpretation, which I think is more on the next slide. So let's hop to the next slide. I think I'm jumping ahead. Yeah. So the outcomes, they organize them across three main stages. So identification, curation, and classification, which I'm sure is familiar to all of us, that sounds like home. So that breakdown kind of mirrors how many of us think about the workflow in practice. First you identify and you prioritize, then you gather, figure out your evidence, and then you classify using your established framework or guidelines. Um, so to the right, I do have just the schematic that they use to show that particular framework, and I thought it was nice that they provided. So they do have 16 learning outcomes that I show on a different slide, but they were able to display how they went from the foundational theory at the very bottom and started from 1 through 5. These are learning outcomes, and then they kind of built up based on what the stage of the curation process was and what the actual task that was being completed was, which I thought was very nice to kind of tie that to learning outcomes. And I thought it made it easier for them to go back and assess where there may have been actual gaps in the people who were taking the training. Um, okay, next slide. Okay, I really like this slide. So a huge picture. However, this was apologies for not having the supplement figure number on here. I must have accidentally cut it off, but this was in the supplement. And manuscripts like this, especially I've noticed in educational manuscripts, sometimes they have really fun and, well, fun to me, but useful information hit in the supplement. And, because when you think of, you read this type of paper and you say, oh, training course curriculum, you say, well, where's the curriculum? But, it's a publication, you can only have so many pages. But I thought this was very nice, and I'm interested to see, um, even if you guys just type it in the chat, like, I'm not sure if you guys have taken a structured course, or maybe even if some of these types of courses or topics or things that you haven't interacted with, that would be fun for us to kind of talk about and, popcorn around about later on at the end towards the panel discussion. But I did think they did a really great job with developing like the key header topics in the dark blue, and making sure they had great introduction, and then going through the different stages of how to filter your variants, collect the data, and different softwares that were used. I also really love that they broke it down into theories, practical modules, and case studies. I think that's really important, especially with training. I know specifically for Gen, like with some of our trainings, we do have like some online case models and things, which I personally found helpful when I joined Gen. They also had a really nice, I thought this was nice, trio sequencing. Some people, I've interacted with some people who aren't even aware what that is, which is, it's really important, I think, for people to know. And even with functional studies, you don't necessarily always have a functional study, but if you do have one, it's important to know, like, hey, what do I do with this type of information, and what do I need to pull out of this? So yeah, I'll leave this for more discussion because I know we do have to have the panel. So let's move on. Let's next slide. I'll keep talking. Okay, so this is just a breakdown of the Bloom's taxonomy that they use. So this was the 16 outcomes that they use for their learning outcomes. So in other words, the framework is not just listing tasks, it's classifying the level of like understanding at different stages of professional development. So I'm going to go through the figure really quick, and then we'll move to the next slide. So they had the categories at the top right, whereas remember, understand, apply, analyze, evaluate, and create. Um, and then they just kind of said, okay, well, if it's shaded, that means like this is introductory, but if we have a dot, you should be more advanced and you should be able to analyze. So on the next slide, though, it actually shows you how they took that framework and revised it to the different career stages that they believe are applicable to a variant interpretation career path. So they believe that students should be remembering, and I really like that they applied active verbiage to it under it. So students should be able to define and find, information about whatever variant. If you are a recent graduate, you should be able to describe, summarize, et cetera, et cetera. I did think this is really nice. I would love to know at the end, like, how do you guys identify? Like, have any of you, do you think you fit into these verbiages that they're using? Like, do you think you can do these things? Or do you think that's like, maybe that's something I should, marinate on and focus on later? Next slide. So now I'm going to get into the results and wrap it up so we can actually have the panel discussion. So the authors reported a significant gain in learner understanding across all of their interpretation learning objectives. So the introductory learners reached like role appropriate proficiency, which kind of suggests that the trainings were effective even for those who are earlier in their developmental stages. And remember, they use that Kirkpatrick framework. So they also determined, based on those 16 objectives, they are saying that 96% of the learners reported an increase of understanding. which is a strong signal of like perceived learning impact. So it means that the training did have some impact on the people who took it. Um, I also thought it was worth noting that a lot of the evaluation is like it relies on self-reported understanding. So it is useful, but it's not the same as a direct performance assessment. Next slide. So I'm not going to go through this entire image. However, this was in the supplement and there is a lot there to unpack, but I did like that they had the supplemental image separated into profession, experience, and I noticed in the chat someone may have put how much, years of experience they had, and your experience with variant interpretation. So I really think this slide reinforces 2 points, scale and adaptability. So they had 951 learners over a 2-year period, which is really substantial. The learner pool seemed very diverse in regards to like career stage and career type. So it really suggests that the framework is like profession oriented and it's appropriate, and it can help support different entry points into this career path. Um, Also, the emphasis on having people's like feedback on their experience with variant interpretation as well as like applying it with those learning objectives. Um, I really think that is going to help people overall in making sure that this type of framework can be used, in other spaces and places. Next slide. I'm not going to go through these images. However, I thought these images were really important because it focused on the confidence, focuses on tool confidence. So specifically increasing confidence in tools like Clinvar, Nomad, and IGB, which are really important to variant curation. So I thought that this practical, like kind of understanding of the tools was really important because it was both conceptual and tool mediated. So it's one thing to kind of understand like evidence categories as a theory, but it's another to like actually go in, retrieve it, assess it, and integrate it, into commonly used resources. So I do think that this training really strengthened some of the users' confidence in using these tools, which will ultimately improve efficiency and reduce variation caused by like people who are unfamiliar with the tools or either using the tools inconsistently. Um, okay, next slide. Uh, yeah, so this is also going over a tool, so we can skip over this one. Okay, so we're gonna just hop into why this matters so we can have enough time to discuss. So, um, I think the main message is that the learning outcomes framework helps define what competence in variant interpretation actually looks like. And it matters because without having a shared framework, training could be inconsistent, assessment can be subjective, and career development can be poorly defined. And I do think, even with this group with the Variant Scientists Network, I think it's really nice because career development is kind of like a little piece of it. So it's important to kind of note that people want to learn, people care about this, but on the back end of it, when we all turn our computers off at 5 or 6 or whatever time, you want to have your career settled, you want to be comfortable in where you are in your space. So I think stuff like this where people are articulating those benchmark outcomes from novice to expert, and it gives you like a clear roadmap to standardize the education. It allows people flexibility in their training format and gives them a larger opportunity to be able to learn in this particular, career space. The authors, they also suggested like their framework could support credentialing and career ladders. I think that's plausible, which is really relevant, especially, as this particular workforce expands. Um, let's see. I think I have one more slide. Do I have another
I think
slide?
that's it.
That's it. Okay, that's it.
That's
All
it.
right. So yeah. So I think this manuscript makes a really important contribution to, you know, shifting the conversation on how we classify variants to how we train people. Um, so I would love to hear some of you guys' thoughts, opinions, questions. I feel like there are probably things in the chat I just hadn't seen. So I'm gonna mute and look at the chat first.
I think, Barbara, you're going to lead the next section of the kind of Q&A.
Okay. All right. So thank you for that wonderful run through of the paper. So our next part is going to be our panel discussion. So maybe each of our panelists could just unmute for 1 minute and give a 1-minute introduction, what their role is and how they ended up as a variant scientist.
I'll go first. My name's Kevin Bowling. I am an associate professor at Washington University in St. Louis. My role here is a little unique, so I'm definitely involved in a lot of translational genomics research work. I also oversee our team of 3 clinical variant scientists in the clinical genomics laboratory. And then I would also kind of call myself a variant scientist by trade. So I've been working in variant science since about 2013. Just reading through this paper, I wish that these resources had been available then. I think that it would have really been helpful for me to understand kind of like, you know, what the knowledge base was, what I should know going into this specific field. But yeah, I work with the variant scientist team at WashU here really closely and look forward to more discussion.
Okay, I can go next. My name is Ashley Birch, and I am an ABMGG boarded molecular geneticist. I'm also the director of the variant science team at GeneDx. And in previous roles, I led groups of variant scientists. So I've kind of seen this from different perspectives, from smaller academic institutions to larger clinical commercial laboratories. And I just kind of got into it just through just naturally. About 10 years ago, we started hiring our first variant scientists, and I began working with them to help build our curriculum, and then just kind of went from there.
And I don't see Brian on the call today, so I think that's it.
Oh, okay. So I guess my first question I would love to hear from both of you is, when a new variant scientist applies to work for either of your labs, how do you go about assessing their skills, and what skills are you looking for in a new hire, let's say at an entry-level position?
Okay, I can start. So coming from a large commercial lab where we are at now, we do have quite an established training program within our company for variant scientists. So we are less concerned about the curation skills that they come in with, because we know we can train those. We are more concerned with their base level of knowledge in human genetics and in clinical genetics. We are concerned with how critically they can assess the literature. So we tend to ask questions around their experience with the literature and what they might take from different studies. And we also want to assess their ability to take feedback, because with an established training program, there's a lot of feedback that goes on there. In previous roles where we didn't have such a established training program, then at that point we might want to see what knowledge are they going to bring and kind of help build our team with, right? So that's kind of going back a bit more years, but we would maybe ask them to explain what they currently do in their roles and how they do it, and what pain points they see, and just understand how they talk about their experiences, and that helps you kind of gauge where their level of knowledge is.
Okay. Kevin?
Yeah, I kind of echo what Ashley said there. I mean, I think us being at a small academic institute like Washington University, at least having a team of 3 variant scientists, and actually When I got to WashU, we had 0 variant scientists. We had some people that were kind of working in the laboratory in that role, but we didn't have a specific position. So we kind of had to build it from scratch, which was tricky. So I would say now even our training is not specifically well defined, but we do have a framework in place. In terms of looking for a variant scientist, I think, going back to what Ashley said, I think knowing that they are thoughtful and logical in their thinking. They have a human genetics background that will allow them to assess the literature in a relevant way. I also think, too, that it's important to find somebody that's adaptable, right? So they don't get kind of caught in doing the same thing all the time and don't want to change their ways, because we work in a field that is evolving super quickly. And then I also look for someone with soft skills, too, right? That can interact with clinical laboratory directors and other colleagues in a very respectful manner, and just bring a positivity to the team that kind of facilitates, a positive training environment, and also just, positive day-to-day interactions.
So thinking about those training programs, and I've also heard from some of the students that I have trained that have gone on to different settings, either like companies with very formal training programs or smaller institutions with more customized training. What would you think the balance is between just like understanding the published guidelines versus understanding like workflows that are specific to your lab or institution? Maybe I'll throw that to Ashley first.
I would say that they're very tightly entwined at a company that has a large training program like we do, where we don't necessarily train and say, well, this is, the published guidelines and here's our, internal adaptation, right? It's kind of tied in together. And I'm sure that every single lab has their own modifications, their own specific workflows, especially if the lab's been around for a while. A lot of the guidelines that have come out, well, when the ACMG guidelines came out in 2015, there was a lot of gray space, right? And labs were kind of filling in the gaps. And then maybe modifications came out, but maybe you weren't able to pivot and adapt those because you'd already adapted yours. And, so every lab kind of has gone their own way a little bit. And so that's something just always to be aware of when you are going from one place to another, because there are some changes, that you may need to make in the way that you approach things or the way that you handle things, that maybe you didn't have to. It was just different, different in different labs. So like I said, I think at least from my perspective, the internal modifications and the published guidelines they're so entwined, you almost don't know where one starts and where the other begins unless you do that background research to try to read those papers and break them out. And that can be part of I think the challenge, because you may not, if you go to another lab, then you've got it so ingrained because they were trained together that you maybe don't quite understand what parts to leave behind and what parts to bring with you. So yeah.
Yeah, I completely agree with Ashley. Our laboratory guidelines are really intertwined too. I should say our modified guidelines are really deeply intertwined with, what's put out from the professional societies. I would say one of the things that we try to highlight too is just, resource access. We know that there's lots of different guidelines that exist now, right? Depending on variant type. And then also we have, even gene-specific guidelines or pathway-specific guidelines. And so just knowing where that information exists and how to get to it and, application of that is something that we try to stress. And then also too, Ashley can probably speak to this even more than I, given the large test menu at GeneDx, but we also have quite a large test menu. So just making sure that people are up to speed with the SOPs and nuances of testing, et cetera, I think is really important for new people coming in.
Mm-hmm. Yeah, so let's talk about the big training thing that I think about, which is the new V4 guidelines are coming. What are some plans to get the existing workforce in your organizations up to speed on how things are going to change? Maybe start with Kevin this time.
Oh, goodness. It's a good question, Barbara. I mean, we've been thinking about that and planning for it, and we've had several members of our variant scientist team that are involved in, helping going on some of the ACM, I should say, the cleansing calls and things that are discussing those, right? So thinking about what that implementation looks like. I should say that our laboratory, we haven't done anything formally yet to really introduce those. I mean, part of that also is working closely with our software analysis system vendor, right, to think about incorporation. So it's very much a work in progress, but we've certainly started as a team thinking about what the new guidelines will look like. And of course, going out and trying to access resources that are kind of a heads up for what's to come.
Yeah, I think I would say pretty similar. It's not something that I think we've had a lot of discussion on yet. We were involved in the recent pilot, and that was really interesting, really eye-opening. But I think there's still more that remains to be seen on what the final decisions kind of end up being, and how we're going to be able to put them into our workflow before we can think about training. So.
Yeah. And I did want to just welcome, I think Brian is now on the call. I'm one of our other panelists.
I am so sorry. This completely
okay.
fell through my email.
That's okay. Maybe if you could just give a 1-minute introduction of your role, and then I'll start just have you answer the first question, which I posed, which was like, what does training look like in your company?
Yeah, so I am the supervisor of variant science at Genomics. So doing mostly exclusively somatic work. And most of my day is actually doing variant science, despite my title. I'd say within neogenomics, most of our training ends up being very sort of on the job and catered for whoever is coming in. So one of the big things that I'm looking at, since we're kind of a more medium-sized lab than some of the larger ones out there, a lot of times we hire one person at a time, and it ends up being very tailored. So if somebody has previous germline experience, we're going to show how to translate germline experience into somatic. If somebody comes in with relatively no experience, which is honestly pretty rare at our team because we don't have a structured training program right now. It ends up being a lot longer of a training session. So that's a very good non-answer, just to say that we kind of tailor things based on what our incoming variant scientists have as a baseline.
Excellent. So in the chat, there's been a lot of discussion about training programs and training opportunities. And for those, I didn't introduce myself. I am a program director for a Master of Science in Human Genetics and Genomic Data Analytics that was designed to prepare variant scientists for this role. But there is a lot of diversity in job ads, what the required education is. So maybe starting with Ashley, what do you think is, maybe just say what your the recent hires you've been involved in, are they at a PhD level, an MS level? Is it genetic counseling? Is it human genetics? And where do we think the field is going to go in the future?
Yeah, I think that every lab is different with the requirements that they look for. When I've been involved in hiring, it's typically PhD level individuals. I would say that most, but not all, don't have formal experience in variant science. A lot of them are coming out of their PhD, or they're coming out of postdocs, or they've been doing more translational research, and they're interested in getting into this field. And because we have, a very well-defined training program, it's that works for us. But I know that a lot of labs are different and have different requirements. But yeah, I would say for, again, most of our recent trainees, that's what we have. Certainly, some people do come in with variant science interpretation experience, and I would say, kind of unlike Brian, we don't really tailor our training, to those people, because our training is, it's a mix of guidelines plus, internal workflows and stuff, that it's all, very similar.
Anyone else want to jump in?
Yeah, I'd be happy to jump in as well. I've kind of had experience as a hiring manager both at Natera and with NeoGenomics as well, and had very different sort of experiences as to what we were sort of looking for. When I was working with Natera, me and one of my colleagues kind of created a very structured training program. So we hired in a lot of people who had very limited experience in variant science, but had uh, either a either a master's degree very tailored for genetic testing or genetic counseling, or PhD level scientists. And just as a plug for Barbara, we've hired a few of her graduates at Natera, and we're very happy with the level of experience and knowledge that they had coming in. But then we'd also hire people who had, five 5 years of experience in variant science, which honestly in our field is a lot because it is relatively new. Um, but then kind of transitioning over to neogenomics, we were focused a lot more on hiring PhD level variant scientists, mainly because at least in the somatic space, there's a lot more focus on functional studies. And as many of you may know, there's a large gap between what a good functional study is and what a publishable functional study is. So we really rely heavily on people with wet lab experience to sort of know that difference and be able to see where the weaknesses are in assays like that.
Yeah, and I would just add to that, Barbara. I completely agree with both Brian and Ashley. And I also think, too, it really just depends on kind of the state of your lab at the time of the hire, right? I mean, when I got to WashU, we immediately needed someone to come in with lots of experience, and we were so lucky to recruit Alexa. And right now, all of our team has PhDs. We actually have one individual that has PhD and MD, which is really helpful for clinical correlation. But I think our team is at a place now where we could bring someone in, maybe that doesn't have a PhD, right, but has that knowledge base and train them up to do the position. So I think it kind of ebbs and flows with the evolution of the laboratory, exactly what you're looking for. But I think that there is a space in the clinical lab and in variant interpretation science for people from a variety of different backgrounds, PhD or non-PhD.
And I'd also like to add on to that sort of there has been a large growth in these academic affiliated hospitals in variant science. And I think that is a great place to start looking if you don't have the sort of baseline experience, because there really has been a contraction within the commercial industry with some major labs being bought out or going under. And I really think a lot of that has to do with this sort of expansion of I don't want to say smaller, but of academic affiliated hospitals where the work is going to be very similar, but you're also going to be closer to a community. You're going to be more involved in sort of the actual patient interaction because you're going to have access to those data, whereas somebody like me might only have the information that's on a requisition sheet. And I've never worked at a academic hospital, so I don't know exactly how that works. But from my understanding, it's another very interesting and very accessible way to get into the field.
Mm-hmm. So most of the time when we think about variant curation workshops and things, we're usually thinking about the single nucleotide variant guidelines for germline interpretation. But of course, there are other guidelines for copy number variants and structural variants, and also for the cancer curation guidelines versus the somatic versus germline. So what advice would you have for a student trying to decide if they want to specialize in maybe not germline single nucleotide variant, how to get training in that? And is it important to have knowledge across all of those domains, or is it okay to start with just the single nucleotide germline and build a career from there? Maybe we'll start this one with Ashley.
Yeah, so I mean, in my experience, as you said, most of the training I think is, more available with SNVs. And I think that for most people, it would, they'd be able to start there and move on and then eventually develop more expertise. I don't have any experience with somatic, so I can't really give any advice at all regarding somatic. With CNVs, I definitely think there's a subset of people who have experience in cytogenetics and, really just understanding how chromosomes work and all the different ways that they can, be a variant. And I feel like that information can be, really helpful. But again, I don't know how one would go about getting that unless they were actually working in a cytogenetics lab, which again, there's just not that many people doing it. So in my experience, I think that typically starting with SNVs is a great place to start. You get the basics, you get exposure to probably just a very wide range of things related to variant science and can specialize from there. But yeah, I mean, that's just, yeah, what I've seen.
Brian, maybe you
I'll
could jump in. Or
yeah, I'll jump in and just say that I would recommend starting with the SNV side of things, mainly because there is a lot of, I don't want to say a lot, but there's overlap between that and copy number variants or any of these large structural variants, and ultimately the guidance around them were kind of slow to develop. So there's a lot of variability lab to lab in how people handle copy number variants, and also just in general in the germline space outside of cytogenetics, those tend to be pretty rare. My only experiences have been involved in the DMD gene or the HBA gene in at the germline level. Cancer kind of becomes the Wild West because all sorts of weird stuff happen. And ultimately, I can't really recommend any guidance for how to get experience or exposure into that type of work. Oh, and the other one that I would like to add is fusions as well, because it's very rare to find people who have experience in gene fusions. But that kind of falls into cytogenetics as well. So I would recommend if anybody wanted to get a little bit of background into copy number variants to try to look at the Duchenne's muscular dystrophy research, just because there's a lot of literature out there on the DMD gene and which exons are important, which ones aren't. And I think that would be the best place to get any background, but I wouldn't necessarily recommend it as something that's necessary for entrance into the variant science field.
So we do have lots of chat, also asking one question that I know in the Variant Science Network we've talked about before, but do you think it would help to have an accreditation or a license in variant science, and what might that look like in the future? And I'll start with Kevin.
Yeah, I think it would be really helpful. I mean, I think it would be helpful for us as hiring managers, probably you all, Barbara, as people that are training this workforce, right? And then also too, just going back to the specific paper that was presented, I think one of the things that they really missed in this manuscript was just some training related to regulatory issues, right? I think that's a huge thing in clinical laboratories. And so I think even having that, at least an understanding be part of the certification would be helpful. But yeah, I think it would be helpful, from the obvious things that we discuss all the time, right? So that training programs know what to cover, so that hiring managers know that people are qualified, and then also so that people that are interested in variant scientists know what they need to do to get to that place. So I think it would be helpful in a lot of different ways. When I was reading through the manuscript, thinking about standardization of this field, I think that the guidelines not only have helped us interpret variants right in a more standardized way, but it really has helped contribute to some of the learning outcomes that were mentioned in the paper, right? So I think that that's come a long way in terms of standardization, but actually having some sort of certification just so that we know that people are competent for these roles would certainly be beneficial.
Yeah,
Ashley, your thoughts? Oh, good.
Yeah. No, I know it definitely knows it's come up in conversation. And I guess I have more questions than answers really around, what it might look like. So, where does one get this training, right? If it's at a program that's, for example, trains people who have masters, then what about, a lab that maybe looks for someone with a PhD? Would they have, the requirements? And then also, how like, how do we balance the number of people who want to get into variant science with the number of people that, a number of roles that we have? Like, I know when I hire, I typically get a lot of interest, right? And so how do we we don't want to have too many people getting, let's say, accredited, but then there's not enough jobs for them, right? We want to make sure that that's balanced. I think that would be something to be careful about. But otherwise, I definitely think it would be really interesting, to know that the people that we're interviewing have, that like a certain level of kind of agreed upon competency. That would be definitely a big change.
I'm actually going to play devil's advocate here a little bit because one of the things I kind of struggle with in the fields that I used to be a clinical laboratory scientist before I got into this field, and there is just a myriad of certifications that people feel like they need to get, and it tends to block people out who don't necessarily have a strong financial footing. And so I'm always a little bit hesitant to require a certification from somebody who's already gone through, 2 to 5 to 7 years of grad school, and ultimately I do think that there is some benefit, but I also think it's important for us to recognize that the variant science field is pretty small. There's not many people in at least the U.S. is the only market I'm aware of that are doing this particular career. So I don't know if necessarily putting the effort into developing a certification would be something that would be a long-term benefit for a field that's still relatively small.
So I want to just piggyback on something you said, Brian. We are talking mostly about what's going on in the United States. Do we feel like there is, I know there are guidelines for Europe, there are other countries making their own guidelines. Do we think this is something that might be unified around the world, or are there always going to be differences where maybe if you get your training in one country, it might not be as applicable to labs in those countries? Or is the internet and the shared tools the great denominator that is going to make the process pretty standardized around the world? Just wondered on your thoughts.
The reason that I'm laughing is I've got a little bit of insight into the process of what ACMG went through to get their guidelines out and are currently going through to get the new guidelines out. And I think that there are so many people with so many big feelings about how to do variant science that I don't think that, in the next decade or so, there's ever going to be a globally unified process for variant science. But I think that ultimately over time, we're going to develop so that all of those processes are very similar. And ultimately, like, I think the European guidelines do a very good job in some places where the ACMG do tend to fail. And I know that there are clinical labs out here that are using European guidelines for certain variants or certain types. So I think it's ultimately helpful to have multiple different points of view. And plus, different parts of the world are going to have different conditions that they tend to see just because of population bottlenecks or net migration. And ultimately, I think it is important to have sort of niche areas where people do focus in. I'm thinking primarily of like Finnish Europeans, or I know that there's certain areas within Iran where there are population bottlenecks that tend to show unique genetic clusters that we necessarily don't have access to.
Fun. All right, well, we do just have 3 minutes left, so maybe we can give each of our panelists 1 minute to share a final thought on education and where it's going to go in the next 2 years, and some of our advice for the people on the call who are obviously interested in getting into the field. And I'll start with Kevin.
Yeah, so this has been a great discussion. Thank you guys for facilitating this. I would just quickly say to people that are in the chat that are interested in variant scientists, I think it's a number of people. Feel free to cold call, reach out to people that you think that may have the knowledge. I also completely agree with what Brian said. I think that kind of academic institutes are a great place. If you are in a PhD program now and you're doing a little bit of variant science as part of your project, feel free to reach out to the clinical laboratory on campus and, ask to meet with some of the clinical directors or people that are doing variant science to get a better idea of it. And also that could be a great way of kind of getting a foot in the door for some training or for some opportunity. So, but great discussion. Thank you, guys.
I think that those are great points. And I'm just looking at, a particular question in the chat, that says, are there any skills or credentials that hiring managers are looking for? How much do you value, continuing education from other companies? I mean, it just, from a personal perspective, I feel like when anyone can show that they have an interest in variant science, that they're not just, applying to a job because it's remote and it sounds nice, but like they've shown that they've taken courses that apply to ACMG. They've worked with a lab, even just to analyze a specific variant, related to their project. They've volunteered for ClinGen, and it was a big one if you have any opportunity, but like anything that really shows that this is really, a real interest of yours, I think helps make someone stand out. And there's a lot of interest in this field. And so, yeah, really just any way to, even small pieces of experience can, be highlighted in your CV, I think.
Yeah, and ultimately, I think too, I'm just going to piggyback off of Kevin because I've already spoken a lot, but the cold calling does really help. Actually, how I got into this position was reaching out to Alexa when I first started, and she was able to help me out a lot. Unfortunately, it tends to be a little bit of good old American know-who in the field, and but that being said, everybody I've ever talked to in the field has been very pleasant and very welcoming to talk to. And yeah.
Right.
Oh, also one more thing, if you do ever have any interest in any specific genetic disease, a lot of smaller nonprofits are looking for people who might be interested in volunteering to do genetic work or to help patients kind of understand the testing a little bit better. And that's another good way to get in. My foot in the door was also nonprofit genetic testing as well. So there are opportunities out there.
Excellent. Well, thank you to our three panelists, and thank you for our wonderful journal discussion to start off today. And I also want to thank everyone in the chat. I know there's been lots of questions and answers, so thanks for making it a very community-oriented conversation today. And I will turn it back over to Alexa. I don't know if we have our next event to announce.
Thank you so much, Barbara. We actually don't have our next event to announce yet, but if anybody has any ideas for topics or subjects or would like to volunteer to present something in May, please reach out to me. I would love to hear it. And thank you, everybody. Thanks to the panel members. Thanks to Jasmine for presenting the Journal Club article as well. Take care, and we will send out our next event invitation soon, hopefully.
Thank you.
Bye, everybody.
Thank you, everyone.